Great. Okay. I think it's starting to slow down. So I think we can kick things off right now. We'll get our opening remarks done and we'll wait for people to trickle in while that's happening. Um, so hello and welcome everyone to the first of many, I hope, Future Fields webinar on our EntoEngine. We're thrilled to finally present a deeper look at how we do what we do. And that is, how do we manufacture recombinant proteins at scale with fruit flies? So today we're in for a bit of a treat. We've got Matt Anderson-Barron. He's our co-founder, CEO, and the inventor of our system. He's going to walk us through the nitty-gritty details of our approach, which has gone pretty much from an innovation on a napkin to kilogram scale manufacturing tech in a little over 4 years. Accompanying him today, we've got some other panelists from our team. We have Ela Dudek. She's our VP of R&D, decades plus protein purification expert here. We have Avi Sheshachalam, our VP of Product. He's our resident drug discovery at Cell Bio Whiz, hosting us, doing all the back-end work, and the creative brains behind our marketing efforts is Diane Jun, our marketing lead, and moderating will be myself, Chris Fetterly. I'm the VP of Sales and Marketing here. I'll read off a couple minor housekeeping notes for the webinar today, and then we'll launch into it proper. Please note, chat is disabled. This is intentional. It's to allow all of our attendees some uninterrupted focus time on the technical aspects of the talk today. For questions, please use the Q&A box at any time during the talk. We will try to get them answered and looked after during the Q&A section of our talk. Speaking of which, we've got about 35 minutes, including these remarks and the presentation, a couple questions afterwards, and then we've got 25 minutes for the Q&A proper. And lastly, a final reminder that we are recording this webinar, so please take note. Lastly, if you or others in your network were not able to attend today, please let us know. We are hoping to do a second presentation, perhaps sometime in the new year, after everyone comes back rested and recharged. So at that, I guess I will stop blabbing. Matt, I'm going to pass it over to you to walk us through the agenda and kick things off.
Awesome. Thanks, Chris. Good morning, good afternoon, good evening, depending on where you're dialing in from. And welcome, everyone. I'm really thrilled to have this opportunity to unveil the behind the scenes of our platform technology, the EntoEngine. So here's everything that we're going to cover in today's webinar. So we'll start with a brief introduction of the company, the EntoEngine, and ultimately why we chose fruit flies as our manufacturing platform. Then we will get into the process, just how the EntoEngine works with the behind the scenes look at how we do what we do, which will segue nicely into highlighting some of the key technical and non-technical advantages of the EntoEngine. And lastly, we'll wrap up with some Q&A from the audience. And of course, feel free to populate the Q&A box throughout the talk. So first, I want to start with our mission, which is to synergize nature and technology to improve life on Earth. This is what we are working towards. At Future Fields, our work has always been motivated by impact, and we believe the best way we are equipped to do this is by leveraging biology and biotechnology. And so this journey to make an impact and the story of the EntoEngine began about 5 years ago when we were working to solve the challenge of high cost growth factors in the cellular agriculture and cultivated meat industry. And it was in this process that we quickly realized that the field of recombinant protein production had reached a plateau. And this was limited by the existing methodologies and expression systems. So this was a really good forcing function for us to think from first principles and ask ourselves, if we could build the best possible protein expression platform from scratch, what would it look like? What features would it have? And then, and only then, would we go out and find the right organism to work with. And this approach is what led us to develop the EntoEngine. It wasn't about just improving biomanufacturing, but rather reinventing it. So what exactly is the EntoEngine? The EntoEngine is our proprietary biomanufacturing platform that utilizes genetically engineered fruit flies, more specifically Drosophila melanogaster, to produce recombinant proteins. So traditionally, recombinant proteins are produced in genetically engineered microorganisms or cell lines. And in particular, 3 different systems produce the bulk of recombinant proteins on the market today. And those three are E. coli, yeast, and CHOK1 cells. Those microorganisms or cell lines are then grown in these large fermentation tanks called bioreactors, which is essentially what we sought to remove from the equation. It's this costly, slow to scale infrastructure that really hamstrings current biomanufacturing technology. So instead of cells or microbes grown in bioreactors, we genetically engineer stable strains of Drosophila that we can express our protein of interest, which can then be cheaply and efficiently reared at large scale to produce the protein of interest. Essentially, we're replacing the bioreactor with an insect. But why fruit flies or Drosophila specifically? Well, the answer lies in over a century of scientific research. This tiny organism has been a cornerstone in genetic studies for 120 years. And this has generated a wealth of genetic tools and understanding of Drosophila physiology. In fact, there are 3 Drosophila stock centers around the world that collect, curate, maintain, and distribute over 75,000 different genetic strains each. So this amazing organism has a deep, deep scientific foundation that's been built upon for over a century now. Now, combine this with the innate efficiency of insects and Drosophila in particular. So first, they have short life cycles, just 11 to 12 days. They have very high fecundity, so a given female lays about 100 eggs per day. Also, Drosophila thrive around 25 degrees Celsius, so that makes conditions for rearing them very easily attainable. And most impressively, they have a remarkable feed to biomass conversion ratio. So in our process, we see about a 2.1 to 1 ratio, whereby just 2.1 kilograms of dry feed is converted into 1 kilogram of dried biomass. So it's this combination of scientific pedigree and biological efficiency that makes Drosophila an ideal choice for biomanufacturing of proteins. So how does this work? This is the first half of our biomanufacturing process, which I'll initially breeze through because I'm going to double click on each of these steps afterward. So for the development of a new strain, it all starts with design of a plasmid tailored to your protein of interest. That plasmid containing your gene of interest is then microinjected into Drosophila embryos. And this ultimately generates flies with that gene of interest stably integrated into the genome. We'd then screen each strain to confirm expression. We can do that by qPCR or Western blot. And once that's confirmed, we'd scale up those strains in our proprietary rearing system. We harvest larvae, so we would harvest the larvae and homogenize them mechanically, and the resulting lysate is then clarified using simple steps like filtration. From there, everything downstream is pretty much copy and paste from other expression systems. Our clarified lysate is then ready for purification. We use column chromatography. With that, we've been able to consistently achieve really high purities, well over 95%. And from there, the purified product is lyophilized for enhanced stability, and the final product will always undergo rigorous in-house quality control to ensure that every batch meets spec. Now, let's zoom in on the first half of our process. So first, creating our stable transgenic strains. So again, this is done by microinjection of the plasmid into Drosophila embryos. And it's at this step, at the embryonic stage, that the gene becomes stably integrated into the genome of the Drosophila. And as you can imagine, this is done with a really, really small needle, which sounds really challenging, but it's actually a process that's been done for decades. It's relatively straightforward, and there's actually a remarkable amount of control in this step. So we know exactly where to insert the gene in the Drosophila genome. So that is, we can actually target integration of the gene of interest into very specific predetermined hotspots in the genome where we know that expression will be optimal. Now, how do we know that the gene actually gets integrated in this step? Well, we do this by pairing our gene of interest to visual markers like genes that control for eye color. So we can actually select for flies where we know integration is successful. So basically, we can inject into embryos from white-eyed flies, and we screen for red-eyed flies in the resulting progeny. And so you can see in the middle image here just how easy it is to distinguish between those two populations. And so these are very specific advantages of the Drosophila system. Now, important to note, everything I'm talking about, everything I'm describing in this process till now is a one-time thing. So generating a stable expression strain only needs to be done once. So microinjection is a one-time thing. So once we have those stable strains, we can effectively rear them in perpetuity. And we can monitor this over time. We can monitor the genetic stability of our strains by utilizing these visual phenotypes. So again, we know in our process that with these strains, the red eye color is linked to insertion of our gene of interest. So in our expression strains, as we're rearing them, we can monitor for any occurrence of white eyes, which would suggest a contaminated stock, and we can rapidly correct for that. So now these stable strains are then scaled up in our novel rearing system. So scale up in traditional biomanufacturing systems is often fraught with challenges and very significant costs. So in those systems, once a master cell bank is established, those strains must be continually optimized over and over again in these subsequently larger vessels, which creates a really fragile process and that's prone to failure. And so because of that, scale up of a process for a given product can in itself take months or even years for traditional systems. Now, in contrast, the EntoEngine scaling process is simple and robust, and that's what we designed it for. So first, again, there's really high fecundity in Drosophila. A single female Drosophila can lay up to 100 eggs per day. So that allows us to exponentially increase populations because basically we can increase a given population 2 orders of magnitude every day. So contrast that with cell systems, which at best have 8 to 12-hour doubling times. Most CHO systems have a full 24-hour doubling time. So while CHO-K1 will double their population in 24 hours, Drosophila are effectively 100xing their population in 24 hours. So it's really remarkable just how good insects and Drosophila in particular are at making more of themselves. And now this fecundity is further accelerated with our novel rearing process. So once a given strain is established, it's scaled up from vials to jars to trays over the course of a couple of weeks. Then once a population reaches these trays, they can be readily and easily maintained at the scale, basically in perpetuity. So you can see what those trays look like in the image on the right here. So with these trays, we can rapidly scale up or down as needed. Production is very flexible, and that's because it's a modular system, which means we maintain smaller, discrete populations rather than one very large population. So that gives us more control over the process, but it has other advantages. It actually prevents the spread of disease. And so with that, our insect team takes really great care to keep these populations healthy and happy. The feedstock is prepared daily under near sterile conditions, and populations are constantly monitored for any sign of illness. Now, the way our rearing process works is each population is reared for 24 hours on fresh food, which is poured into the bottom of each tray. You can see the yellowish looking material in each tray in the image there. And so that feedstock is a proprietary blend of ours, but it contains agarose, which makes it into a firm gelatinous-like substance. The adults then eat, mate, reproduce in those trays over the course of a 24-hour period. And so the females will lay their eggs directly into the food. And after 24 hours, the food is basically saturated with eggs. And this is something that our team determined empirically and have optimized for the right population size for a given surface area of those trays. And so at 24 hours, those adults are removed from the tray and then given a new tray with fresh feedstock. And it's not because they consume it all, but again, because they saturate that surface area. Remember that females will lay up to 100 eggs per day. So once those trays have been conditioned with the eggs, they're moved into a separate chamber to hatch into larvae, where they then spend the next 3 days basically eating nonstop. They consume the entirety of the food and turn it into biomass. So at the end of day 4, they look something like the tray on the bottom of the slide here, where basically the entirety of the feedstock is consumed and it's turned into larvae. And it's at that point that they are harvested, which is simply done with water and a mesh screen. And so this whole cycle of seeding trays and maintaining these populations is actually remarkably simple and easy to do, which was always by design. Now, because this process is cyclical on a 24-hour cycle, it allows us to harvest and therefore manufacture continuously. And this is a feat that's really unparalleled in the biomanufacturing industry. It is truly continuous production. We're the only system in the world, as far as we're aware, that can do this continuously, daily, in perpetuity with minimal resources. So once the larvae are harvested, they're stored in a frozen state. And depending on the protein of interest, this can actually be done for months at a time. We've been able to build up an inventory of frozen biomass that's been stored for months, and then we can still take that, process it, and purify fully functioning protein from it. And with our process, it's really easy to track every batch of biomass to the exact population. So we have full traceability in our process. And this is particularly important to note. We're not operating with GMP standards at the moment, but we fully intend to in the near future. And this aspect, the ability to trace our starting material back to each specific lot, is really important part of enabling GMP standards. So how do we go from frozen larvae to purified protein? Well, the next step is processing. Essentially, the biomass that's frozen is thawed. We run it through a mechanical homogenization step. This disrupts the tissue and creates a lysate. Now, importantly here is that our process doesn't require harsh detergents or chemicals to lyse the cells, which is often used in other expression systems. And that's because most often the protein of interest can be extracted directly from this raw homogenized lysate, which means we can employ gentler methods that don't disrupt or unfold the protein. We're not dealing with things like inclusion bodies. And that ultimately leads to a better functioning protein. So after mechanical homogenization, the lysates are readily clarified with centrifugation, filtration steps, to develop this clarified, transparent looking liquid. You can see in the middle, looks a lot like apple juice. So you can see just how clean we can get the lysate from a few simple steps. But just because the steps are simple, this process was not so simple to develop. Our team painstakingly developed some really innovative methods to make this process extremely efficient. Now, because of how efficient it is, we can already process tens of kilograms of biomass per day at just a benchtop scale right now. From there, everything downstream is more or less the same as other expression systems. So we utilize column chromatography for purification, plus some other steps like TFF to concentrate the final product, followed by lyophilization for storage. And again, importantly, is we can achieve ultra-high purity with our process. The EntoEngine is novel, but it's still compatible with all the downstream off-the-shelf solutions for protein biochemistry and purification. So now I want to highlight a few specific technical advantages of the EntoEngine that also contribute to making it such a great manufacturing platform, which are tunable expression of your protein of interest, tissue-specific expression by utilizing a wide number of cell types found in Drosophila, genetic stability of our strains, and the ability to produce highly functional complex proteins by facilitating all the proper processing and folding steps required. And we'll dig into each of these in more detail right now. So again, there's a lot of genetic tools that have been developed specifically for the Drosophila system. Like I said, over 120 years of studying them. So generally, our goal at Future Fields as a manufacturing platform is to boost expression. We want to get the highest titers we can. And that's something that we have been working on and can continue to improve on. So already by implementing some new genetic tools into our process, we've been able to demonstrate major expression boosts with our products. So for example, we saw a 10 to 14x increase in expression of our FGF2 with our newest production strains compared to our original strains. And importantly, higher expression means lower cost of goods. So those are directly correlated. But it also means consistency. We have predictability in our manufacturing because of the uniformity of our strains and the ability to tune expression as needed. But importantly, and important to note is that we've really just begun to scratch the surface of what's possible with our genetic toolkit. There's so much more opportunity in the genetic tools, so many cool things that we can continue to utilize and iterate on that will have profound effects on our proteins and their manufacturability. So insect cells are really good at producing complex proteins. We've shown this with a number of proteins, but so have others in academic research settings. So for example, there's a publication shown here on this slide that demonstrated the expression in Drosophila of functional human SERT. This is a transmembrane protein with 7 transmembrane domains. And one of the reasons that the EntoEngine is particularly good at this, at producing complex proteins, is the optionality we have. We effectively have tens of different cell types to choose from, all being grown at scale with the exact same inputs. And so this, in my opinion, is one of the coolest and most interesting aspects of what we do, of our platform. We have neurons being grown at scale, epithelial cells, gut cells, muscle cells, all being produced at scale, all produced with the exact same inputs. But importantly, we also have the tools to target expression of a transgene or protein to any one of those cell types. We can readily employ tissue-restricted expression in cases where a protein of interest requires a particular cellular environment for proper folding and processing. And this is extremely unique in the manufacturing landscape and something that I think really sets the EntoEngine apart. One of the major challenges currently facing biomanufacturing is genetic stability. So cell lines for biologics, most commonly CHOK1, these are immortalized and they're prone to accumulation of unchecked genetic mutations, and that can really affect the output. It can even affect the efficacy of the protein of interest. Now, on the contrary, our process employs stable genetic strains where successive generations always stably inherit the transgene of interest. So again, by utilizing many of the unique features of the Drosophila system, like visual phenotypic markers, we can maintain and monitor the genetic stability of our strains in real time. But we can also leverage something that we haven't talked about yet, which is balancer chromosomes. So balancer chromosomes basically prevent multiple alleles on the same chromosome from being separated during meiotic recombination. They effectively prevent recombination events. And the reason that's important here is to prevent the loss of your transgene over subsequent generations. So balancer chromosomes are by far the most commonly employed in the Drosophila system, largely because of the ability to pair them with visual markers that can be used to track them. So similar to eye color, we can use other visual markers like the curly wing phenotype that you can see in the bottom left of the slide here. So while not impossible to use balancers in other systems, it's really cumbersome and limited. But in Drosophila, we can readily and easily employ them for all of our strains, which we already do. We use balancer chromosomes in all of our production strains, and that allows us to generate these remarkably stable engineered strains. So for example, on the agarose gel in the Western blot on the right-hand side here, you can see expression from 3 generations of our prolactin strain. And as you can see, expression doesn't really change at all. And that's something we've observed with all of our production strains over relatively long periods of time. So this aspect, genetic stability, is really important in the development of novel biologics. So with the EntoEngine, the strain that you use for process development is the same strain used in manufacturing, which is really unique. So if you're a biopharma company working on a novel therapeutic, that means you would have the same strain for preclinical studies as you would for everything downstream, including clinical trials and commercial launch. It's the same strain for the lifetime of the product development lifecycle. And that's something very unique, and it comes with tremendous advantages with predictability, with regulatory filings, and that sort of thing. So one of the things that makes insect cells really great protein factories is their ability to facilitate all the key processing steps that are required for a number of different proteins. So for example, furin site processing in the TGF superfamily of proteins is essential for activity. And we've shown the EntoEngine's ability to facilitate these processing steps with both TGF beta 1 and activin A, as you can see on these 2 Western blots. So production of this superfamily of proteins can be quite challenging in other systems because they all undergo multiple steps of critical processing in vivo. So they are synthesized as large precursors that are subsequently cleaved in 2 steps to yield the mature protein homodimers. And that cleavage step is facilitated by the furin protease, which is inside the Golgi complex. So without proper furin site processing, these steps don't occur. And since these proteins are only active as dimers, this process is really critical for their biological activity. And again, as you can see, expression of both of these, TGF beta 1 and activin A, in the EntoEngine shows the presence of the processed, fully mature protein in both cases. Now, the best part about all this is that these benefits are available to you. The EntoEngine's for everyone, and as a result, anyone can take advantage of these benefits. As we were developing our platform, we realized the benefit that our expression system could bring to the manufacturing landscape. So in September of this year, we officially launched our CDMO service. So we're offering the EntoEngine to you through our custom protein manufacturing services. So whatever your needs are, whatever the application, we can optimize production of your target protein to help you hit your milestones faster and more affordably. So our expression system is novel, but our approach to quality certainly is not. Bleeding edge doesn't have to be risky, and that's because we're committed to the highest quality standards. We have been from the very beginning. One of our earliest hires was for quality, and we now have a fantastic quality team that ensures our production process produces the highest quality material. We already use pharma compliance software, including our EQMS, and we have flexible bioanalytical tests to meet your needs. And our team of in-house regulatory experts are here to help with every project, regardless of the application. Now, lastly, I want to end on something that's really important to us. Sustainability is really central to everything that we do at Future Fields. And this has ultimately been the driving force behind what we do since day one. The EntoEngine inherently promotes sustainable biomanufacturing, but beyond that, we're committed to sustainability in our day-to-day operations. We've invested in a number of sustainable initiatives, including 1% for the planet, Migraine Lab certification, APT labels for our reagents. This is an ethos that's embodied by the entire team at Future Fields, and we're really passionate about sustainability in biotech. And with that, I want to thank you all for joining me today, and thanks for your attention. I hope you learned a little bit more about the EntoEngine. If you're interested in exploring this technology further, please feel free to reach out to myself. We have our general contact listed here. So thank you again. More than happy to answer any questions alongside Ela and Avi, and I will pass it back to Chris now to facilitate the Q&A.
Great. Thanks, Matt. Thanks a lot. It's always exciting listening to you talk and nerd out a bit about our system. Very cool. So if I got all the panelists back on the camera, yeah, this is great. So we do have a couple questions to sort of set the tone, if you will, of the Q&A. So I'll ask those because they often come my way as in sales and marketing. So we'll address those, and that might help seed some ideas for questions that you may or may not have. So the first one is going to be towards Ela, because you're in the protein world. Can you talk to us about what happens if there are protein host interactions, if we're expressing our proteins, and how that's managed?
Yes, so because we have so many tools to work with, which has been developed by other scientists, and we just have to repurpose them to tune to our needs. And we can work with strong or weak promoter. We've actually had on our pipeline a case where when we overexpress the protein, we compromise the viability of our flies. So we quickly changed the tool to use weaker promoter, and now we have viable strain which can rear and we can grow at scale. And we have other ones which we are about to employ, or to repurpose from academic world and bring to our lab. So we can talk more, but I think I will give others room to speak.
Very cool. Thanks, Ela, for addressing that. And feel free to ask more questions on host proteins if you also choose. Avi, we get this one a fair amount as well from potential customers and beyond. How do we, because our system's continuous, how do we define our lots if we're not using discrete bioreactors? What does that look like?
Sure, I mean, that's a great question, and it's a very common question for all continuous bioprocesses, and even FDA is taking note of this. From a regulatory perspective, a batch or a lot can be a certain predefined quantity of product or a quantity of product produced in a certain predefined time interval. So there's significant freedom for the end user, that is the client, to determine what this flexible batch looks like. What our quality control and quality assurance personnel can do for you is determine the limits and help you understand, based on risk mitigation strategies, risk assessment, what a lot or a sublot can look like, and combine that with our efficiency of our uniform batch size or uniform stably expressing biomass, and use this filter of flexible batch sizes. You can have a unique system that works with evolving needs of manufacturing requirements for clients of any size.
Cool, thanks, Avi. And then our last kind of pre-thought question to get the creative juices going. And this actually seems like it's related to your question, Tarika, as well. So I'll ask both of them combined. So Matt, we often do get this question. Can you give us a sense on how to think about yield with respect to the EntoEngine? And then Tarika's specific question in the Q&A was, as a C. elegans nerd, I was easily able to follow processing steps in the EntoEngine platform, wondering if you get better titers compared to yeast or other expression systems. So they're trying to think about yield with our system.
Yeah, it's a really common question. People like to ask it, I think, because then they can root it and compare it to something they're familiar with. It's hard to answer. It's hard to do a direct apples to apples comparison here. And that's because of the way we measure titers. In yeast systems or E. coli or CHO, they're typically measuring that milligrams or grams per liter. So the size of your bioreactor tank, you know, if you have 1,000-liter tank and you get, you know, a kilogram yield, you have a 1 gram per liter titer, and that's how they would measure it. With us, it's a little bit different because it's in mass. So, you know, we measure in milligrams per kilogram of biomass. So it's a bit of a challenge to do that apples to apples comparison because we're looking at raw biomass volumes. That being said, what we like to do is just root it in cost and scalability. So, you know, your production per, you know, milligrams per dollar, that sort of thing, or dollars per milligram. And typically we see much better results with our system, even at a benchtop scale with where we're at right now. We did outsource a TEA about 2 years ago to a UK service provider. And even at that time with our system, we came in about 16 to 20% cheaper than Pichia Pastoris. And our process has obviously gotten a lot better since then. So yeah, at the end of the day, we just like to root it in cost. And I'd say typically we come out on top.
Very cool. Thanks a lot. That's our pre-planned questions. I'm going to jump into all the Q&As that are coming in now from the audience. Matt, I think this is an appropriate follow-on for you. This one's from Gareth. He asked, is the 1 kilogram protein production that you mentioned in 60 days, is that the raw biomass or is that the final protein target?
Yeah, so to clarify, that is the actual final purified protein target. And that comes back to, again, just the raw efficiency and scalability of the insect system. So to clarify, that's if we took a given strain, so we had a you know, some threshold quantity of our strain, and all we did was scale it up, you know, allow it to reproduce and scale it in our system for 60 days. At the 60-day mark, we could then employ a manufacturing process where we could produce 1 kilogram per day. So that means 1 kilogram on day 60, 1 kilogram on day 61, 1 kilogram on day 62, so on and so forth. And we can maintain that in perpetuity. And again, that's just highlighting the scalability advantage we have with our system.
Cool. Thanks, Matt. Hope that answers your question, Gareth. I think this is a good one for Ela. Ela, Andrew asked, can your process be engineered to create lactoferrin?
Of course it can.
Okay.
To be more specific, we've researched this protein already, and we know that it's possible to express the protein, functional protein in Drosophila, and you know what we can offer, customized approach to lactoferrin or any protein you have in mind, and we can offer inducible expression, you know, targeting different hotspots. As I said, strong weak promoters. We have plenty of tools to play with to boost expression and to maintain expression, and also, you know, without compromising the activity and functionality of the protein. So yes, lactoferrin, I would love to have on our pipeline if possible, cool. And I would love to try new tools we can test with this protein.
Awesome. Avi, I saw you nodding your head quite a bit. Did you want to add anything on lactoferrin briefly? You're the product roadmap person. So.
Yeah, the Ela kind of talked about researching this protein. We've actually expressed transferrin, which is a closely related protein that essentially does the same function as lactoferrin, which is an iron binding carrier. And we were able to prove that it can be produced, and it's a holotransferrin. So it already comes with the iron bound in the protein, and it comes out as red in color. So it's a pretty good test case for our system.
Very cool. Thank you. And maybe Avi, while I got you, because you've got sort of the holistic view across the entire platform, can you comment on which part of the optimization for our system was the most challenging from your point of view? And then maybe Ela and Matt, you can chime in as well, because we probably all think different parts were very challenging. But Avi, let's start with you.
I think the most fun part, let's not call it challenging, was the scale up. You know, because just the idea of growing these fruit flies at the scale that Matt's just talked about was something that I couldn't wrap my head around a few years ago. And now we're able to produce several kilograms of that biomass so easily. And combine that with genetic tools and process development tools, and the existing downstream processing that we have done and the quality control that we have, I think we have generated this unique system that beats any scale that has had 3-4 decades of research in the past, and we've done that in the short 2-3 years. So that, I think, is something that I'm so proud of, and it's been the funnest part of our process.
Great. Ela, any questions on comments on optimization? What was challenging or fun?
Okay, so since I was very early on and
Yeah.
I started at the bench, I can speak from my perspective, and I think it was actually extraction process, because
Okay.
we utilize completely different tools than other industries, which I was very familiar with. Yes, and it's so easy, so simple, but yet so discovery for me that again, repurposing something for our process, and it took us quite few months to actually find this this tune this to work towards our needs. So I say that extraction.
Extraction. Gotcha. And Matt, really quickly, do you have one you want to add? And then I'll move on to some other questions here.
Yeah, it was getting the funding to do it. That was the hardest part.
Yeah. Yeah. Okay.
I'm biased, but that's my comment. But yeah, everyone's gonna have a different answer depending on where they're working. But I would just comment that all of it has been due to some pretty incredibly creative and innovative individuals working on this platform. So
yeah, definitely, I echo that. Can you quickly comment, Ela? Shrish Harsha asked, is the purification tag removed in the final protein product? I'm guessing this varies depending on the protein we're working with here.
It's per customer. If you we have capabilities, of course, to remove a tag. We have couple enzymes working with proven that we can do this. And yes, but if you prefer to leave a tag, also possible. Yeah.
Gotcha. Thank you. Oh, this is a good one for you, Avi. I'm going to ask this one here from Daniel. Great presentation. Thank you very much. Have you performed, or are you planning to quantitate the immune stimulatory potential of your growth factors? This information will be essential, especially when working with patient-derived cells like PBMC. Can you comment on how we're maybe thinking about immune response, or potentially looking at immune response in the future?
Absolutely. This is a great question. And actually, it goes all the way to preclinical applications in animal models as well. So you don't want to have unmitigated allergenic response to your protein. Oftentimes, this is determined by downstream process. The purity of the protein also determines allergenic potential. What we've seen is, for the proteins that we have and currently in our pipeline that are up and coming, we always go for 95% and above purity. And this kind of reduces significantly what we can expect from immune responses. Having said that, we are in the process of seeing animal data very soon, hopefully. So that will also help us understand not just growth factors and cytokines, but also any and all proteins that come out of this platform and how they can be suited for therapeutic applications in the future.
Very cool. Thank you, Avi. Ela, we got another one. Everyone likes to drill in the nitty-gritty of these proteins. It's awesome. Do you confirm proper folding of the proteins? How do we think about protein folding? And is it the client's responsibility to do that potentially? What are your thoughts there? And Avi, feel free to add afterwards.
So of course it depends on the client. Some clients prefer to do it themselves, but also we do have some capabilities to confirm that. We do have a quite robust cell team with we can test the function, and we also have SPRs of plasmon resonance, and we can study through this, and we're open to learn other techniques, but also it's very, very custom based approach. But so far all the proteins we we're able to produce purified. They are all functional, and we haven't struggled with the functionality. So folding.
Anything to add there, Avi, or should I truck along? No. Okay, cool. Okay, that's this one. Avi, maybe let's jump to this one, and then maybe Matt, you can add on here since we just had a pretty cool in-house talk all about this. This is anonymous attendee asks, you mentioned GMP compliance for therapeutic proteins. Can you explain in some detail? Explain the same in some detail. So I guess they're asking about how we think about compliance and our general quality approach in GMP.
Yeah, there are several parts to this answer. First is traceability and transparency. I think that is the key for good manufacturing practice. So as kind of Matt mentioned, every larvae that we produce coming from a stable stock is tracked. And we are able to make sure that that tracked information is documented using an infrastructure that is compliant with CFR Part 8 of it. And this infrastructure includes EQMS system, ELN systems, and others. And we are able to generate the right kind of regulatory paperwork to enable IND filing. So when it comes to GMP, there is the document management system, but there's training of the right personnel and tracking the training and also the process itself. Because GMP is very protein process specific. And we are, as and when R&D teams are able to generate the process for a given protein, and the manufacturing teams take over production of this protein, we are able to generate the paperwork, the batch production records, the master batch records, the equipment management, so on and so forth, that allows us to start becoming more GMP compliant. So when are we GMP compliant? That's an important question. I think that that is based on our pilot scale manufacturing plan that's up and coming. When we do the engineering runs, we'll have the necessary paperwork and other documentation to fully be transparent for different clients to audit us and give them the necessary confidence that our process can give them full traceability. Anything from you, Matt?
I think you covered most of it, Avi. I would just add as a sentiment, our team has done a really good job of creating a solid quality foundation that will allow us to move into GMP manufacturing processes pretty seamlessly. And a lot of that's just, like I said, because of the foundation that's been created. So while we're not compliant yet, it's not going to be an insurmountable challenge for us, or even anything that we can't do readily because of this early investment in quality at the company level.
Awesome. Thanks, Matt. Thanks, Avi. This is really cool. What we got here. So probably your world, Ela, again. For proteins that have homologs in Drosophila, are there overexpression linked issues with organism viability, post-translation modification, purification? We somewhat addressed that, but if you want to talk about it a little bit more, overexpression linked issues. And then the second part to this question, would proteins from other non-bovine human species be viable using your system?
Yes, so addressing the first part, we haven't experienced yet the problems with homologs, even though there are orthogonal proteins in Drosophila. We the one I mentioned before, which overexpression was causing issues with viability of Drosophila. It was completely different protein, human one, but sometimes when we see some issues, we can address this immediately by changing the strain. But crossing the strain with so far weaker promoters always were helpful. So that's addressing this. And the other part of the question was.
Sorry, I was on mute. The other part of the question was, would proteins from other non-bovine, non-human species be viable in our system? So they're kind of probing or what are the limitations for species types expressed in our system?
Yes, so yes, they are viable, and they are already, being expressed by other researchers. And there is also on our pipes we have two which are from prokaryotic cells. So yeah.
Very cool. Awesome. Thank you. Oh, Avi, what's up? Question or comment?
Well, not just other species, completely brand new proteins. I mean, think about the kind of changes that are occurring in the protein field with AlphaFold and others coming out. Now we can do chimeric proteins from pieces from every different species that we know, and that can be produced easily in our system. Non-species protein.
Very cool. Awesome. Thank you. Yeah, so a couple of questions on PTMs. Maybe I'll start with you, and then maybe Avi or Matt, you can add on to this because I know we've got some projects on the go specifically looking at PTM. So the first one is from LaShern. Thank you, Future Fields, aside from the presentation. I'm curious to know if your EntoEngine expression system is able to faithfully reproduce the post-translational modifications, e.g. glycosylation, as in mammalian cell expression systems, and if so, does your QC process validate this for each protein produced?
Yes, so we know from literature that Drosophila is able to do some glycosylation, and it has the same enzymes as mammalians. There is less of them, so Drosophila is not able to do those finest glycosylation patterns, but the simple ones, yes. And which we differ from yeast, for instance. Drosophila doesn't force glycosylation on the proteins that they are not meant to be glycosylated, and I find this being very a huge advantage of our system compared to yeasts. And we know that we are lacking sialic acid glycosylation. This like a cherry on a cake for mammalian systems, but of course, we're trying to address this. We just built the whole program, and we want to create a fly which will have all necessary humanized enzymes introduced to Drosophila. And in, I think, like 2 years mark, we might have this strain available, and we'll be testing how this machinery of fully humanized glycosylation work in our system.
Very cool. Matt, did you want to add any comments about PTMs? Because I know we think a lot about PTMs.
Yeah, I mean, Ella already alluded to it, but just something that we're really excited about is this project we call GlycoFly, a bit of a nod to the previous GlycoFly. But this is an exciting project. It's fully funded now. We're moving forward on this starting January 1st. But as Ella said, we'll be able to create a background strain that produces fully humanized glycosylation patterns that we can then use for manufacturing identical human proteins for therapeutic applications, which is really exciting. It opens the door to a lot of new opportunities that we can start to get into. So yeah, pretty excited about the future with that one.
Cool. A little teaser on the GlycoFly project from Future Fields. Thanks. Rich, I think we answered your question about post-translational modifications. So I'll mark that as done, but if you feel like we didn't hit it, please just ask a new question. I'll move on to the next one. I think, Avi, this is well suited for you and looks to be our last question in the talk. Anonymous attendee asks, do you see a pathway for injecting into humans? What is the regulatory pathway for that? I'm assuming they mean the recombinant proteins that are expressed, injecting these into humans. Yeah, any comments there?
Yes, there is a precedence already with the FDA and EMA. The European Medical Agency and the FDA in the US, as well as Health Canada, has approved insect cell-derived proteins. Flu vaccine, for example, is made in insects. So is HPV vaccine. And this gives us confidence that as long as we are able to prove safety data, and we are able to show the traceability of our processes, audit the facility from these agencies, we are able to start thinking about therapeutic applications of the products that are emanating from our platform. Ultimately, I think this is where we are heading in the long term. And it's essentially one protein, one product entering into this space will prove that this platform will be able to do many more after that. So from FDA's perspective, from regulatory bodies' perspective, are we safe? That is really the main question that we must address. And we are on our way to generating the right kind of data, keeping in mind the quality and the client's considerations to essentially address this particular point. So there is a regulatory pathway, and we are on it.
Very cool. Thanks, Avi. And I think that's the end of the questions. That's no more questions. We got 3 minutes to go, so might as well wrap it up here and let people get on to other things in their days. Thank you so much, Matt. Thank you, Avi. Thank you, Ela. Thank you, Diane. Thank you, all of our attendees from around the world who were able to tune in today. It was a real pleasure to speak with you all and teach you more about what's going on at Future Field. So thank you, and we'll see you next time.
Thanks, everyone.